Okay, so thank you for joining this webinar today. So this webinar is mainly going to be a software demo taking you through the main features of PEAKS Studio. All right, so the first slide that I have here shows you the main workflow that is used in PEAKS. So PEAKS is able to read in your raw data, and the first thing that it does before doing any other analysis is Denovo sequencing. And the reason why we do this is that it actually feeds in to all the other parts of the analysis. So I'm going to take you through Denovo sequencing and talk a little bit about what makes a good Denovo sequencing result. And then I'm going to go through and talk about the different identification algorithms that are used in PEAKS. So the main protein identification algorithm that we have is the PEAKS-DB search. So this is a Denovo-assisted database search that uses Denovo sequencing to make it more accurate and sensitive. Then I'm going to talk about PTM identification with PEAKS PTM. So this also uses Denovo sequencing in a way to identify PTMs that you may not have considered in your initial assumptions about your data set. And then I'm going to talk a little bit about SPIDER, which is actually a Denovo-based homology search. And the idea here is that it's able to link up your Denovo sequencing results to the database that you search against in order to find single amino acid variants. And once you do all these searches, whenever you do an identification search with PEAKS, you always get a list of Denovo-only peptides, and I'll talk a little bit about that. So that's actually a list of the Denovo sequences from scans that cannot be matched by your initial database search. And that's where you can find some really useful extra information that may not be possible to find with the database search. All right, so just a quick slide here. So I designed this slide to give you an idea of how PEAKS Denovo sequencing works under the hood in a very general way. So PEAKS has a unique algorithm for Denovo sequencing that uses dynamic programming and machine learning to predict the most likely peptide sequence for a spectrum without even looking at a database. And it's actually able to do this very quickly with the use of dynamic programming and machine learning. So to give you an idea of how PEAKS works in one direction, you start from your precursor mass in a spectrum, and the assumption here is that you know where the next peak is going to be if you're looking for an amino acid peak. So you know that it's going to be within a certain mass range from your precursor mass, and then PEAKS will look in that range and try to figure out which ion is most likely to be from the next amino acid in the sequence. And then moving on from there, it looks at the next mass range where you're most likely to find an amino acid, assigns one, and then continues on, and actually does this in both directions at the same time in order to prevent conflicts between B and Y ions, in this case assuming that we're working with a CID or an HCD spectrum. And this is what it does with every spectrum that you give it in your raw file. And it's able to do this very quickly. On a standard desktop PC, it's able to do it at a rate of about 15 spectra per second. All right, so the next general principle behind PEAKS Denovo sequencing is once we actually predict the most likely Denovo sequences for every spectrum, it will actually generate thousands of possible candidates and then score them. It will then assign local confidence scores to the amino acids in the most likely de novo sequences for any spectrum. So what it does with this is, let's take an example here. So this is a sequence that was assigned to a spectrum, and it uses the concept of permutation, meaning how close of a fit is this match compared to all other possible matches. So say, for example, we look at one individual part of the sequence at a time. So it looks at individual chunks of the sequence one at a time and compares it to the different other possibilities. So let's say, for example, we look at this EAD, and then we compare it to the other possibilities at this position with the same mass. And if the other possibilities are a very poor match to the spectrum, then we can assume that actual, so the original match was ADE, then we compared it to EAD and other possible matches. If those are bad matches to the actual peaks in the spectrum, then we can assume that ADE is the most likely match for that sequence, and then we give it a red color here, meaning that we have over 90% confidence that those are the correct amino acids for that position. And it will continue on through and do that for all of the different chunks of that peptide sequence to figure out what the kind of confidence score we can actually assign to that de novo sequencing result. All right, so let's take a look at how that works in the software here. So here we have your raw de novo sequencing result. And with every set of data that is run through PEAKS, you'll always get this de novo sequencing result. And if you click on this de novo tab, it will show you the most likely match to every spectrum that is given in your data file, so long as a reasonably confident match could be found. So a question that often gets asked is, what actually makes a good quality de novo sequencing result? So what kind of score cutoff should we use? And people often will use an ALC score, which is the average local confidence here, as a cutoff to determine whether or not a de novo sequencing result is accurate or not. But the thing is you need to consider more than just the average local confidence when looking at a de novo sequencing result. So that's why we have this button up here in the top left where you can actually set a local confidence cutoff. So if you go here and click this button, it will show you the color coding system for our local confidence score. So amino acids that are above 90% confidence will appear red. Anything between 80 and 90% confidence will appear purple. Anything between 60 and 80% confidence will be blue. And anything less than that will be black. So what I'm going to do here is I'm going to go set a local confidence cutoff of 80% and set this tag length to sort by tag length here. And what this tag length actually means is it's the longest string of amino acids that are above 80% confidence. And that's actually controlled by this here. So if you set this slider here to 80%, then the tag length will require that confidence be at least 80% or more. If you set this to, let's say, 90%, then it will require that the confidence be at least 90%. And you'll notice here that the tag length for these sequences has changed because we're setting a more stringent cutoff. And if you go back, you'll notice that it went from 31 to 32 here. So what makes a good quality de novo sequencing result? So you can see here for this peptide sequence that we have 32 amino acids in a row that have above 80% confidence. So that's actually a very good de novo sequencing result. And you can see why here when you actually look at the peptide spectrum match. So how peptide spectrum matches are shown in PEAKS, any matchup between an ion and an actual peak in the spectrum that is from an X, Y, or Z ion will appear blue. Anything that's from an A, B, or C ion will, anything from an X, Y, or Z ion will appear red. Anything from an A, B, or C ion will appear in blue here. And you can actually control which ions will actually be shown in your spectrum over here with this wrench. And you can see the default settings for CID, ETD, or even ETHCD here. And you can control whether or not certain ions will appear or not. So you can restrict it to just B or Y ions, change anything that you'd like here, or just reset it to default. Yeah. So that's the ideal de novo sequencing result. So I'm going to show you a little bit now about why we actually say that you need to look at the local confidence and not just consider the average local confidence. So if you look at result that's more middle of the road, say a de novo sequencing result with a tag length of 7, meaning that 7 amino acids in a row are above 80% confidence, such as this one here, you can see that, yeah, we do have really good confidence in the middle part of this peptide sequence, and that's what the most common result is for de novo sequencing, because the middle part of the spectrum is going to give you the most information that you can use to actually determine an amino acid sequence. Then the lower mass regions and the higher mass regions are much harder to actually get a de novo sequencing result for. So that's why you actually need to consider the local confidence when looking at a de novo sequencing result. As you can see here, we can't actually get ions that are showing fragmentation between the E, P, and G and the low or the high mass region, so we can't actually get the B or the Y ions that distinguish between these amino acids. So PEAKS is actually letting you know that really all we can do is guess at the amino acid sequence there. We know that there's some sort of amino acid sequence that's 509.2 Daltons, but we can't be entirely sure what that is. We can give you the most likely guess, but it's telling you here by the low local confidence of those amino acids that there are other possibilities that fill in that gap. And you can actually see that here. If you click this All Candidates button, it will show you, let me just lower down the confidence slider here, it will show you the top 5 sequences that match up to that spectrum, and you can see that in all of those top 5 sequences, the amino acids that remain constant are the ones with the highest local confidence. And that will be true in most cases. All right, so that's a short overview of de novo sequencing. Some other additions that we have in more recent versions of PEAKS is you'll notice that you have this area column. And this can be useful in giving you an idea of the abundance of certain peptide features in comparison to others. And we actually have this area column in all of our identification modules of PEAKS. So this can be used for a few things, such as PTM profiling, and give you some semi-quantitative features. And I'll quickly show you what this area actually comes from. So what you can do in any result of PEAKS, you can actually right-click on the peptide table and say Show Spectrum in LCMS. It'll actually jump to this LCMS heat map view, showing you, if you zoom out, it shows you the full signal of everything coming out of the mass spectrometer, and it will highlight in red the actual MSMS spectrum that matched up to this de novo sequencing result. So if we zoom in on this here, and then you can click the MSMS spectra button here, and it will show you all the MSMS spectra that were found in this data set. Going back here, you can click this Features button in the top right, and it will actually show you the peptide features that matched up to this LCMS information. So what this heat map is showing on the x-axis is the M over Z versus the retention time on the y-axis, and the darker the signal here, the more intense the signal is coming out of the mass spectrometer. And then on the top right, you can switch to a 3D view. It's holding control here to zoom in, and you can get a clear picture of the actual data coming out of the mass spectrometer. And the area highlighted by this shape shows the peptide feature associated with this de novo result. And the idea here is once we actually calculate the area of this peptide feature, this is what's shown in this area column, which gives you an idea of the de novo results coming from the most intense signal coming out of the mass spectrometer, which then gives you an idea of which peptides are actually the most abundant peptides in your data set. So I could do that to do some, use that to do some semi-quantitative analysis, which I'll show you in the other parts of PEAKS. All right, so yeah, just going back to the slides. So that's Denovo sequencing. Moving on to the other parts of PEAKS when we actually infer proteins from that information. So let's talk a little bit about PEAKS-DB, which is our protein identification search. So this figure here shows you the concept of PEAKS-DB. So for every spectrum that's fed into PEAKS-DB, we include the Denovo sequencing result as well as a traditional database search result to in both cases, both Denovo sequencing and database search, the concept is we're trying to find the most likely peptide match to a spectrum. So two different approaches to try to find the same answer. So when you keep that in mind, database search and Denovo sequencing result, if the spectrum is ideal, should come to the same answer. So what we do is we compare the Denovo sequencing result to the database search result. And if that result is, if the database search result and the Denovo sequencing result are very similar to each other, we'll actually increase the confidence in the Denovo sequence, the database search result, sorry. The database search result score will actually be increased. But if the Denovo sequencing and database search result are dissimilar, then the confidence in the database search result will be lowered. And the idea here is we're actually able to separate the true and the false hits much better and make the end database search result more accurate and sensitive by using this approach. Right. And then we also have PEAKS PTM included. So the concept here is that most peptide-centric approaches typically find unmodified high-abundance proteins, and low-abundance modified proteins are usually underrepresented in database search results. And the traditional problem is that in most cases, you can only select a small group of PTMs in one search, because if you search for too many PTMs, it makes the search space too large and it makes your search time too long, or you end up running into memory problems and those sorts of things, and actually just makes the search impossible. Other problems include you may not actually know all the PTMs to select. So the idea behind PEAKS PTM is it actually eliminates the assumption that you know which PTMs are actually found in your data set. Yeah, so the solution is to use PEAKS PTM, which is the combination of Denovo sequencing and database search of only the proteins that were already found to be the most likely proteins in your data set, to limit the search space, which actually allows you to search for all of the naturally occurring PTMs in the Unimod database. All right, now let's talk a little bit about SPIDER. So I made this general overview slide here to give you an idea of what SPIDER does. So what SPIDER does is it takes the Denovo sequencing result as an input. Going back to our discussion about Denovo sequencing, you get an idea of which amino acids in your Denovo sequencing result are the most confident matches to the actual spectrum. So anything that's red is the highest confidence, and other colors are lower confidence than that. So keeping that in mind, it'll actually search the database to try to find a part of the database that is the closest match to your Denovo sequencing result, such as what it found here. So the advantage of searching in this way is that you'll often find that even when if you have a confident single amino acid variant, you'll find parts of the sequence that are not an exact match to what's found in the database. But you can see here that we still had high local confidence in the amino acid that didn't match to the database. So SPIDER will consider all that information and make a call based on what it can see from the database match and the Denovo sequencing match what the most likely amino acid is at that particular position. And is they actually able to find a single amino acid variants and still stay within a 1% FDR and combine those results with your traditional database search results. Yeah, so then it compares your spider result to the actual spectrum, does a peptide spectrum match, and if that's a good match to the spectrum, it will say, yeah, this can be included in the final result. Okay. So the reason why I talked about all of that information together is because when you actually do a search in PEAKS, the PEAKS DB, PEAKS PTM, and spider search results, and even Denovo sequencing can all be set up from the same search space. So here we have in this example here, we have a project where we're just considering 2 data files. So we have a sample which was actually extracted from cells that were in the interphase of development compared to cells that were taken from the mitosis phase of development. And the idea behind this search was we're trying to find the differences in PTMs, specifically focusing mainly on phosphorylation, trying to find differences in PTMs at different stages of the cell cycle. So how we'd actually go about setting this up in PEAKS, when you load the data in, you just want to add the 2 different samples and then specify the enzyme, the instrument, and the fragmentation type. And it's important to keep this information in mind because PEAKS will actually use machine learning based on the instrument and fragmentation type that you put in to determine the most likely ions that you'd expect to see in each of the spectra that are fed into PEAKS. And the enzyme can be used in certain cases. Say, for example, you're loading in different enzyme digests. You can actually combine those different enzyme digests together to get the highest coverage possible. And that wasn't used in this case. In this case, we're just using 2 trypsin digests. Okay, so once you load this into PEAKS, you can click Finish, and once the data is successfully loaded, this data node here will appear solid green, meaning that the data was loaded successfully. And then if you want to search the whole project together, you can click this top button here, then click the PEAKS DB button. So this is the project node, so we click the project node here, then click the PEAKS DB search button, and it will bring up the general search page. And from this page, going back to default, it will actually set up the parameters for PEAKS DB, PEAKS PTM, spider, and Denovo sequencing. So the parameters that you have to set in PEAKS are pretty straightforward. So you set up the error tolerance, the precursor and fragment error tolerance. If you go up to the top right-hand corner and click default, it will give you some recommended parameters for your instrument. In this case, this was Q-exactive data. So we're using, we can use anything in the range of 5 to about 20 ppm for the precursor mass error, and for the fragment ions, usually 0.01 or 0.02 Daltons is acceptable because we have high accuracy also for the MSMS scans. And for the enzyme, you can just select specified by each sample because we set the enzyme when we loaded in the data, and then allow for some non-specific cleavage at one or both ends and the number of miscleavages that you will actually allow within the peptide. For the PTMs, one rule that is important to keep in mind for the PTMs is that it's best to limit the PTMs for just your PEAKS DB search to at most 10 variable PTMs in the basic PEAKS DB search. So in this case, we selected carbamidomethylation, oxidation, phosphorylation, and acetylation, the end term. and then you can include up to 10 variable PTMs there. And then keep in mind here that while we are limited to 10 variable PTMs here in this PeaksDB search in order to limit this, keep the speed manageable of the search, down here in the general options, you can see that we've said find unspecified PTMs with PEAKS PTM. And if you click this advanced settings, you can see by default we're searching 313 built-in modifications, which are all the naturally occurring modifications in the Unimod database. And if you want to limit it to only a few of those naturally occurring PTMs, you can actually just select the preferred modifications, and you can see here that we have a list of all the common, uncommon, glycosylation, artificial, and you can even set customized modifications. And I just want to mention here that the PTMs that are searched in your standard PEAKS PTM search is everything that's found in the common and uncommon lists. Glycosylations as well as single amino acid variants are not included, but you can include those as well if you'd like from this advanced settings options here. All right, and then find more mutations with spider here. That's the spider search that we talked about back in the slides. And once you set all these options here, it will search all those different algorithms here from this one search page. So once that's complete, you'll get a full list here. So you get a Denovo, PeaksDB, PEAKS PTM, and spider node. And to get a full list of the full set of results, you can open the spider node, and this spider node will actually contain results from everything. So once you do that, you can go back to the summary view here, and you'll notice that the first thing that you see is this false discovery rate curve. And this will show you the number of identifications by peptide spectrum match that were found at each false discovery rate. So the x-axis shows the number of peptide spectrum matches, and the y-axis shows the false discovery rate. So as you increase the number of peptide spectrum matches, you can notice that the false discovery rate goes up. And what you can do is you can extrapolate from that by clicking the FDR button up here in the top left and select an FDR cutoff. We have a few presets here. So anything from 0.1% to 5%. And once you click, let's say, for example, we use the most stringent cutoff, it will show you up here in the top left the -10 log P score, which is a score that we use in our identification results. It will show you the -10 log P score where a 0.1% false discovery rate is achieved. And to talk a little bit about the other filters that you can set here in the summary view, you can set a PTM filter. So you can either select an A score or ion intensity. Some recommended parameters there. For the A score, we usually recommend an A score cutoff of 20 or greater is usually a good cutoff for A score. To give you an idea of what the A score is it's a score that calculates the likelihood that a PTM is occurring at the position that's reported in comparison to the other possibilities. And it is also a -10 log of a p-value, similar to our identification result, meaning that a score of 20 is equal to a p-value of 0.01 or less. So that's a less than 1% chance that the PTM is being assigned to the position that's reported. It's a less than 1% chance that the PTM is being assigned to that position by a random chance. Or if you use ion intensity, so that requires that you have matched ions showing fragmentation before and after that modification site of at least, say for example, 5% relative intensity. Or you can set a filter specifically to the mutations, so single amino acid variants as well. And then we also have a protein minus 10 log P recommended cutoff. There is usually 20 or greater, but if you're setting it as identifications to have at least 1 or 2 unique peptides per protein, you should have a minus 10 log P score of 20 or greater if you set the unique peptides cutoff to 2 or more anyway. So the protein minus 10 log P score is not as important as making sure that you assign at least 1 or 2 unique peptides per protein. All right, so let's take a look at, so once we've set all these filters, let's set some filters here and apply those. All of the results that you see from that point on in the protein view will be within those filters that you set. So once you set those filters, you can be confident that the results that you see are accurate. So let's take a look at what we found here. So this is the main view of PEAKS identification results here, where you can see in this view, you can see the protein that we're looking at here, its coverage, the number of peptides, the number of unique peptides, its description, and then you can see the actual coverage view here. Notice that we have these letters with a colored background. So these indicate modified peptides. So we have different phosphorylation sites, acylations, and oxidations appearing here, as well as some other single amino acid variants that I'm not showing it at this moment, but you can take a look at them later. So all of the modifications that are shown here have an A score of at least 20 or greater, so we can be pretty confident in the actual phosphorylation sites that we're seeing here. And some of the new features that have been added in PEAKS 8.5 is you notice that we have coverage for the interphase and the mitosis samples here. So these coverage columns give you an idea of the similarities and differences between two samples. So you can start to get an idea of some semi-quantitative information from this page. So this can be controlled with this button here. In 8.5, you can click this button, and it will show you some different optional columns. So you can show the coverage, or you can show the number of spectral counts, or you can show the total peptide feature area of all the unique peptides there. So you can see here, you can see the total area of all the peptide features for the interphase and the mitosis phase. You can also see the total number of spectral counts comparing the two samples. All right, and if you want to validate any of these results, you can click on any of the peptides here, and it will show you the best spectrum that matches up to that position. You can scroll on the peptide here, and it will show you which ions are associated with which amino acid. So for example, here, let's look at the actual position where we have this phosphorylation at this threonine here. You can zoom in on that position, and you can actually see the ions that are showing fragmentation before and after that amino acid. So we can be pretty confident that we have a correctly assigned phosphorylation here. All right, so once we've gone through, we can be pretty confident in the actual identifications here. So what other kinds of information can we gather from this? So let's consider, we want to ask the question, what are the actual differences in the amount of phosphorylation that are occurring in the interphase and mitosis phase? So you can actually do this with this PTM profiling button. So just to go back here, I click this PTM profiling button, which can be found in any of the views in PEAKS PTM or Spider. You can click that, and it will bring up this extra view here. Up in the top left, you can say show all the modifications. If we are including mutations as well, You can see up in the top left that you could say show all the mutations if you're only interested in the mutations or just the modifications, or you can just look at specific modifications, such as in this case, we're only interested in phosphorylation. And what this will show is it will show you the area of the peptide features if they're found in one sample versus another in terms of the actual position. So this is a phosphorylation at S14, and it will show you the myosin log P, the ion intensity, and it'll show the areas of the modified peptide features, the unmodified peptide features in one sample versus another. And it'll also show you the unmodified and modified at best peptide spectrum matches. So what this allows you to do is, you see here in the bottom left, it shows, in this case, showing the log 10 areas, or you could switch it to percentage. It will give you an idea of the difference in phosphorylation at the different sites. So you can see here at this T39, we're seeing much more phosphorylation in the mitosis phase versus the interphase. And you can see the breakdown at the different positions. And yeah, so this gives you a bit of semi-quantitative information here. If you wanted to go in more depth, you can even add some replicates and see whether or not you see a similar trend across different biological and textile replicates, and it will show you that here as well, and give you a full picture of whether or not you're actually seeing true differences in phosphorylation in different stages of the cell cycle. And this can be applied to any sort of situation where you want to see the differences in PTMs between samples. Yeah, and this can all, up in the top right-hand corner, you can export this as a CSV file, and you can do some further analysis there. So this feature has been greatly improved in PEAKS 8.5. It's really a feature that could truly be used to find differences in modifications between samples. All right, so that's PTM profiling. You also see in the peptide tab, you can do there's similar options here. Up on the top, you can select sample areas or spectral counts to give you an idea of the differences in the peptides between samples. And the last thing that I'd like to show you here is this Denovo only tab. So this is a feature that is truly unique to PEAKS. So going back to the first slide, remember that the workflow of PEAKS is that once the identification is complete, we still get this list of Denovo only peptides, meaning it's a list of the spectra that cannot be matched by any of the identification algorithms that we've used, but still have a good quality Denovo sequencing result. So that will be this list here. And you can still see that we have an ALC filter of 50 or greater, and that we still have 3,452 spectra that still have a good quality Denovo sequencing result above that cutoff. And you'll notice here that these even still have really high local confidence scores at every amino acid in the peptide sequence in many cases. So these are really interesting peptides because they could not be matched by the database search. So that leads you to the question, what is the actual source of these Denovo sequencing results? So these could be PTMs that were not considered in the initial database search. They could be mutations that are longer than a single amino acid variants. It could be from proteins that weren't included in the database. There could be lots of factors that could cause a good spectrum to not be matched. The important thing here is that even though these were not matched by database by database search, you still have this Denovo only tab, which gives you a list of spectra which could be identified if you make some changes to your search parameters or try to identify them by different means. So you could take out these Denovo results, put them into a blast search, anything that you could think of. The important thing here is that we still know which spectra we have that are not identified. So we have a starting point to make some good, true discoveries. All right, and that's all that I had to talk about today. Thanks for taking the time to join this webinar.